Biology 101, Lecture 16. Yay! Last lecture of the class. Today we're going to look at microevolution and macroevolution. So we're going to do it like this. We're going to define macroevolution first. Microevolution consists of changes in allele frequency in a population over time. Macroevolution, however, refers to broad patterns of evolutionary change above the species level. But what is a species? A species is a group of populations whose members can interbreed and produce viable, fertile offspring, but do not breed successfully with members of other populations. And that brings us to reproductive isolation. So gene flow between populations holds the species together genetically. Reproductive isolation is the existence of biological factors or barriers that impede two species from producing viable, fertile offspring. Hybrids are the offspring of crosses between different species. Reproductive isolation can be classified by whether factors act before or after fertilization. Some barriers to reproductive success. They can be prezygotic or postzygotic. Prezygotic barriers block fertilization from occurring by impeding different species from attempting to mate, or preventing the successful completion of mating, or hindering fertilization if mating is indeed successful. Postzygotic barriers prevent the hybrid zygote from developing into a viable, fertile adult because of reduced hybrid viability, reduced hybrid fertility, or hybrid breakdown altogether. And here is all of that represented pictorially. So the prezygotic barriers, there could be habitat isolation, so can't mate. There could be temporal isolation, so they may be in flowering at different times in the year or a month apart or something like that. There could be behavioral isolation, where, for instance, in birds, the mating ritual is very strictly regimented. The songs have to be in a certain order, otherwise it just won't work. And so you see all these mating attempts with a different species. It's just not going to work, and that's why you have that circle with this slash in the center. So this is not going to work. You can have mechanical isolation, where one species just doesn't fit with the other species mechanically. Like beetles or bugs, they have very hard exteriors. They have an external carapace or a skeleton, and it would be very difficult for beetles of different sizes to mate. There could be gametic isolation, where the gametes are just not going to be able to mate because their fertilization is not going to occur. They could be dumped in water at some other time. They just don't get to mate. Postzygotic barriers, we just talked about reduced hybrid viability. Fertilization has occurred now. At this point, fertilization has occurred, but the hybrid is weak, or it's not fertile, or it just breaks down altogether. Now, you may end up with a viable, fertile offspring, but that is almost, the probability of that is very rare. Prezygotic reproductive isolation. So, habitat isolation. Two species can encounter each other rarely or not at all because they occupy different habitats, even though they're not isolated by physical barriers. Temporal isolation. So, those two species breed at different times of the day, different seasons, or different years, and they cannot mix their gametes. Behavioral isolation. Courtship rituals and other behaviors unique to a species are effective barriers to mating. Mechanical isolation. Morphological differences can prevent successful completion of mating. Gametic isolation. Sperm of one species may not be able to fertilize the eggs of another species. It's just not happening. Postzygotic reproductive isolation. So fertilization actually has occurred now between two different species, but you end up with a reduced hybrid viability. What does that mean? That means that genes of the different parent species may interact and impair the hybrid's development or its survival in its environment. Or we could have reduced hybrid fertility. Even if the hybrids are vigorous, they may be sterile, like horses and donkeys produce mules, and the mule can be vigorous, but it's sterile. Or you can have a hybrid breakdown. Some first-generation hybrids are fertile, but when they mate with each other or with either parent species, offspring of the next generation are feeble or they're sterile. Speciation. What is that? Speciation is the evolutionary process by which new species arise. Speciation can occur in two ways: allopatric speciation or sympatric speciation. In allopatric speciation, geographic isolation restricts gene flow between populations. Reproductive isolation may then arise by natural selection or genetic drift or sexual selection in the isolated populations. But even if contact is restored between populations, interbreeding is prevented. In sympatric speciation, a reproductive barrier isolates a subset of a population without geographic separation from the parent species. Sympatric speciation can result from polyploidy, natural selection, or sexual selection. A hybrid zone is a region where members of different species mate and produce hybrids. Hybrids are the result of mating between species with incomplete reproductive barriers. A hybrid zone can occur in a single band where adjacent species meet. For example, two species of toad in the genus Bombina interbreed in a long, narrow hybrid zone. Hybrids often have reduced fitness compared with the parent species. When closely related species meet in a hybrid zone, there are three possible outcomes. First, you could have reinforcement. Second, you could have fusion. And third, you could have stability. So here is a hybrid zone. You can see on the left-hand side, the population is blue, there's gene flow, and then all of a sudden there's this fork. And the population starts to diverge. So the isolated population is diverging and it's becoming a different color. It's becoming pink, whereas the original population continues to become blue. But suddenly there's a hybrid zone, and these two are starting to breed with each other. So what are the possible outcomes? You could have reinforcement, where these hybrid zones are actually reinforced, or you could just have fused them, where the hybrid zone is just closed up again and you get the original population back. Or you get a stability where this continues to happen all the time. The interval between speciation events can range from 4,000 years, as in some cichlids, to 40 million years, as in some beetles, with an average interval of 6.5 million years. So it doesn't take, it's not rapid, it takes quite a nice long amount of time. Macroevolution is the cumulative effect of many speciation and extinction events. So now we're going to look at evolutionary relationships. Phylogeny. Phylogeny is the evolutionary history of a species or a group of related species. The discipline of systematics. It classifies organisms and determines their evolutionary relationships. And from that, we get taxonomy, which is a scientific discipline concerned with classifying and naming organisms. Classification is done in a very systematic way. The two-part scientific name of a species is called a binomial, and it's always italicized, and that's just the convention. The first part of the name is the genus, and is capitalized. The second part, called a specific epithet, is unique for each species within that genus. Linnaeus introduced a system for grouping species in increasingly inclusive categories. The taxonomic groups, from broad to narrow, are domain, kingdom, phylum, class, order, family, genus, and species. And that is what this picture is, which is from your book. So at the very bottom of the picture, you see the three domains, archaea, eukarya, and bacteria. And then in eukarya, you see one branch coming out, kingdom. There are many kingdoms, but one is Animalia. And out of that, there are many branches, but we're looking at just the one, which is the phylum Chordata. And then out of those phyla, we're only picking one class, which is Mammalia. And out of that class, we're only picking one order of many, and that is Carnivora. And out of all the carnivores, there are many families. And we're picking the Felidae family. And then out of that family, there are many genuses, but we're picking the Panthera genus. And then ultimately, we're picking the species. There are many species of Panthera, but we are interested in Panthera pardus, which is the pretty cat sitting on top. We can look at the connection between classification and phylogeny this way too. So we look at the order, which is Carnivora, and then we go to the next step up, and that is the family. And there's Felida, which we just looked at, but there are other families too. There's Mustelida and Canidae, and so that's one branching group. And then out of that family, we can look at what are the genus. So Felida just has one genus, Panthera, but Mustelida has two, Lutra and Taxidea. Canadia has just one, also it's just Canis. But if we look at Canis, it actually bifurcates into two species, Canis latrans, which is the coyote, and Canis lupus, which is the gray wolf. So this is how we can go between classification and phylogeny. If you wanted to construct a phylogenetic tree, it's a very nice way to do it is by building a character table, and that is simply a square. And in those squares, what do? You pick ideas or traits, and you very simply give them yes, they have it, or no, they don't have it. So every no is a 0, and every yes is a 1. And you can start looking. For instance, in this very first row, variable column, does the lancelet have one? He does not. But does the lamprey have it? Yes, he does. Does the bass have it? Yes, he does. Does the frog have it? Yes, he does. And does the turtle have it? Yes, he does. And so does the leopard. So we would say, aha, vertebral column. And if you look on the left-hand side, the right-hand side, the same thing is shown as red in the phylogenetic tree, a red bar. And we look at the next characteristic, hinged jaws. Let's see. Lancelet does not have it. Lamprey does not have it. Bass does. Frog does. Turtle does. And leopard does. And on the right-hand side, you see the green bar, which says hinged jaws. Then we go to pick another characteristic. 4 walking legs. Lamprey doesn't have it. Lancelet doesn't have it. Bass is a fish. Frog does. Turtle does. Leopard does. And you look at the yellow bar, 4 walking legs. And the only things that have those are frogs, turtles, and leopards. And then we look at the amniotic sac. Lancelet doesn't have it, lamprey doesn't have it, bass doesn't have it, frog doesn't have it, but the turtles and leopards do actually have amniotic sacs. And that's the little blue bar. And then the last separation is hair. Lancelet doesn't have it, lamprey doesn't have it, bass, no, frog, no, turtle, no, leopard, yes. So leopard is on his own. He has to be separated out from the turtle because he has hair, but the turtle does not. So you start to separate out these differences, and you realize that there are many commonalities, but there are some differences which are unique. So the terms homology, analogy, orthologous, and paralogous. A similarity can be the result of homology or analogy. Homology is similarity due to shared ancestry. Analogy is not. Analogy is similarity due to convergent evolution. So you could just happen to live in the same environment, and because of the pressures of that niche, evolution will converge, and you end up with those characteristics. So it has nothing to do with your ancestry, but it has everything to do with this environment you're in. Orthologous genes are found in a single copy in the genome and are homologous between species, only diverging after speciation. Humans and mice diverged about 65 million years ago, and 99% of our genes are orthologous, so they're the same. Paralogous genes are a result from gene duplication and can diverge within the clade that carries them and often evolve new functions. Molecular clocks. A molecular clock uses constant rates of evolution in genes to estimate the time of evolutionary change and is calibrated against the fossil record. In orthologous genes, nucleotide substitutions are assumed to be proportional to the time since they last shared a common ancestor. In paralogous genes, nucleotide substitutions are proportional to the time since the genes became duplicated. There are differences in clock speed. If most of the evolutionary change in genes and proteins has no effect on fitness, then the rate of molecular change should be regular like a clock. The differences in clock rate for different genes are a function of the importance of the gene or how critical the specific amino acid is to protein function. There's also, just to muddy the waters, there's horizontal gene transfer. So while we're exulting like, aha, we can find a clock, we can date it, and we can figure it out and reason it out, and we can separate all the beings, all the organisms, actually, no, not so fast, because there is something called horizontal gene transfer. There have been substantial interchanges of genes between organisms in different domains, not just species or genus or family or class or order or any of that. Domains, which make clock speed estimations difficult. Horizontal gene transfer is the movement of genes from one genome to another. Horizontal gene transfer occurs by exchange of transposable elements and plasmids, viral infection, and fusion of organisms. So it's a tangled web of life. On the left-hand side, you see ancestral cell populations, and here is horizontal gene transfer going on. We believe that mitochondria and chloroplasts were indeed the result of horizontal gene transfer from cyanobacteria and proteobacteria. So they're coming into plants and animals and fungus, and that is where then we're kind of stumped as to how to calibrate the clock. Multicellularity. The evolution of eukaryotic cells allowed for a greater range of unicellular forms. A second wave of diversification occurred when multicellularity evolved and gave rise to algae, plants, fungi, and animals. The Cambrian Explosion refers to the sudden appearance of fossils resembling modern animal phyla in the Cambrian period, which is about 535 to 525 million years ago. In the Cambrian Explosion, a few animal phyla appear even earlier. Sponges, cnidarians, and mollusks. They were earlier than that. Fungi, plants, and animals began to colonize land about 500 million years ago. Plant vascular tissue appeared about 420 million years ago. Tetrapods evolved from lobe-finned fishes around 365 million years ago. So you can see where this is headed. Tetrapods showed up like 365 million years ago, so that means colonization of land is coming pretty soon. The human lineage of tetrapods evolved around 6 to 7 million years ago. And that doesn't mean we're that old. It just means that the lineage of tetrapod from where humans originally came from. But the modern humans originated, it depends on various books. Some of them will say 50,000 years, and some of them will say 200,000 years. So modern humans originated, we could safely say, if we're being conservative, 195,000 years ago. Here is a graph which will show you the time at the bottom in millions of years, in exactly how many, 600 million, 500 million, and then the time zones like Proterozoic or Paleozoic, and where these animals actually originated or when they started out. So you can see the sponges and then the cnidarians actually were there before the Cambrian explosion came around. And actually so did the arthropods and the mollusks. They had been around before too. But the echinoderms and the chordates came after the Cambrian explosion. It's not just a Cambrian explosion, there are also mass extinctions. The fossil record shows that most species that have ever lived are now extinct. If the rate of extinction increases dramatically, it is called a mass extinction. In each of the 5 mass extinctions in the last 500 million years, 50% or more of marine species became extinct. The Permian mass extinction, which was about 251 million years ago, and in less than 500,000 years of this period, 96% of marine species went extinct. So it took 500,000 years to get rid of just about all marine life. The Cretaceous mass extinction, which was 665 million years ago, caused the dinosaurs to go extinct, and we remember that as the last mass extinction. Here's a graph of the 5 mass extinctions. So let's just look at it a little carefully. On the x-axis, you can see the time in millions of years. And you can see the eras, Paleozoic, Mesozoic, and Cenozoic. Cenozoic leads to the present. And on the y-axis, you see the total extinction rate. Okay, so what we're looking for is the extinction is in red, and the number of families is in blue. So let's just look first at just the red marks. And you can see 5 peaks. One, which is around 444 million years ago. There's an arrow at the bottom. And one peak around 359 million years ago, a really big one at 251 million years ago. And then comes another one around 200 million years ago. And then the dinosaurs went extinct 65 million years ago. So these are the 5 major extinctions in the last 500 million years. Of course, the Earth is 4.7 billion years old, so we're just looking at the last small chunk, but the most interesting one. And if you look at the blue, so let's look at the number of families. How many things went extinct? Well, in the Paleozoic, the first extinction, I would say some went extinct. And the second, some did go extinct. And in the third, which was 251 million years ago, there was a really big extinction going on. And then the fourth, there were some that went extinct. And when the dinosaurs went extinct, there was a little dip, but that did not mean that all things went extinct. It was mostly dinosaurs and other stuff. The dinosaur extinction is thought to be caused by a huge meteorite that collided with the Earth. And we see the remains of that at the Chicxulub crater, which is in Mexico. It's in the Gulf of Mexico. So we would, and this is just a pictorial depiction of what must have happened. A huge crater was excavated, and dust and dirt and things flew up in this huge ejecta curtain that you see at the bottom. It got lofted up and blocked up the sun's rays. So the rain became acidic, and there was so much that was blocking the sun rays. The plants started to die. The dinosaurs had no food. So they started to die, and so that's how that happened. This is another timeline, but this one is a little bit more complete, and so it goes from left to right. So about 4 billion years ago, 4.6 billion years ago, Earth began. But the first fossils that we know of, it depends on which book you look at, but it's 3.8 billion to 3.5 billion years ago that we saw the first fossils, the first prokaryotes, which are single-celled organisms. And then the first eukaryotes, but still single cells, were only 1.8 billion years ago. So it took a long time, almost 2 billion years for the prokaryotes, for the eukaryotes to evolve after the prokaryotes had evolved. And then once the eukaryotes were evolved, it only took another 1.2 billion years before the first multicellular eukaryotes showed up. So not that long as compared to the first period. And then 500,000 years ago, there was colonization of land by plants, animals, and so on. Oh, and yes, there's the Cambrian Explosion around 525 million years ago, which resulted in the colonization of land. And we are now at the present. The geologic record is divided into Hadean, which is the very old Earth, the beginning of time for Earth. The Archean, which we actually have records of. Hadean, we do not. Proterozoic and Phanerozoic eons. The Phanerozoic eon includes the last 500 million years, and the Phanerozoic eon is divided into 3 eras: the Paleozoic, the Mesozoic, and the Cenozoic, and that's what you probably hear a lot of. Here's a picture of that. So if you look at the eons on the left, the Phanerozoic is the one that's in green, and it's the biggest one. And before that, you can see Proterozoic, Archean, and Hadean. And then if you look at the eras, there are none that we know of for the Proterozoic, because we don't really have complete records of anything. But for Phanerozoic, we do know Paleozoic, Mesozoic, and Cenozoic eras. And then you can divide that time, because this is billions of years. You got to divide it up somehow into periods. And we have the Cambrian period, so that would begin the Phanerozoic eon. And then we keep on going up to the Mesozoic, which starts with Triassic, and then the Cenozoic, which starts after the Cretaceous period ended, and currently we're in the Quaternary period. As for the epochs, because we can still divide this time up, we're in the Holocene epoch, and if you look at the letters, words in parentheses, the Mesozoic era was actually age of reptiles, and the Paleozoic, the blue part, is age of amphibians, and the purplish blue part is the lavender part is age of fishes, and before that, the really pretty purple part is the age of invertebrates, and this is all in the Phanerozoic. But the Precambrian, which is Hadean, Archean, and Proterozoic, that's about 88% of geologic time. So if we look at what do these words actually mean, Holocene, what does that mean? So we can look at it and we can say what was the development going on. So humans developed. The orange part is considered age of mammals. So that's the tertiary period of the Cenozoic era. And you can see the extinction of dinosaurs right around the Cretaceous, the end of the Cretaceous period, which is 65 million years ago. The first flowering plants showed up around 144 million years ago. The first birds showed up about close to 160 million years ago. And then the extinction of trilobites and many other marine animals. That was that big extinction event in 251 million years ago. Large coal swamps. So these occurred because there was a lot of trees, a colonization of land with trees, and they all got felled, and we ended up with coal swamps. So this is all very interesting stuff, and as you can see, this is all in the interesting things are all from Paleozoic to Cenozoic, before that, not much happened except single-celled animals and mostly prokaryotes. This is the same stuff that we just looked at, but it's in a little bit more detail and with pictures to help us remember what happened from the Hadean eon through the Paleozoic era. And this just shows before us what happened in the Cenozoic and what happened in the Mesozoic eras. This is another way of looking at the timeline. Instead of a straight line, you could depict it as a circle. And then you could start with, on the right-hand side, the origin of the solar system, the Earth, and that would be the Hadean period. Then the Archean, and then the Proterozoic, and this is all billions of years ago. And you can start adding colors to show prokaryotes. When did they start? When did atmospheric oxygen start? Close to 2.5 billion years ago. Well, a little bit more than that. And when did single-celled eukaryotes start? About 2-ish billion years ago. When did multicellular eukaryotes start? About a billion years after, half a billion years after that. And then colonization of land didn't occur until recently in the Paleozoic. So we could look at the same timeline in this fashion also. What does the fossil record tell us? Major boundaries between eras correspond to major extinction events in the fossil record. The fossil record is biased in favor of species that existed for a long time, were abundant and widespread, and had hard parts. So we don't really have a complete picture. All we have is some record of some of the organisms that lived in those times. We don't really know what happened because a lot of it just didn't preserve. The oldest known fossils are called stromatolites. These are rocks. They are rocks, and they're formed by the accumulation of sedimentary layers on bacterial mats. And the stromatolites date back to about 3.5 billion years ago, and you can see them off the coast of California. Prokaryotes were the sole inhabitants for more than 1.5 billion years. So they ruled the world. There was nobody else around but them. Here is the addition of oxygen. So about 2.7 billion years ago, oxygen began accumulating in the atmosphere and rusting iron-rich terrestrial rocks. Molecular oxygen produced by oxygenic photosynthesis reacted with dissolved iron and precipitated out to form banded iron formations. So if you look at this picture, you do see striations on the rocks, and that is the iron, the red rusted iron that is produced by oxygen accumulating in the atmosphere and rusting out the rocks. Here is a picture of the rise of atmospheric oxygen. So 4 billion years ago, there was hardly any. There was maybe not even a trace. And then all of a sudden, around 2.7 billion years, oxygen started to rise. And then it abruptly went up, plateaued for a while, and then went up again. And today it's oscillating a little bit, but we're right at almost 20% of oxygen. With the rise of oxygen, many anaerobes went extinct, but others adapted and used cellular respiration to harvest energy. The oldest eukaryotic fossils are 1.8 billion years old. Eukaryotic cells have a nuclear envelope, mitochondria, endoplasmic reticulum, and a cytoskeleton. Mitochondria and plastids, like chloroplasts, were probably small prokaryotes that got subsumed into larger ones as endosymbionts. So plate tectonics. This is another part of the story. It's not just fossils, but what happens to the rocks. So the land masses of the Earth have formed a supercontinent 3 times over in the past 1.5 billion years. One happened about 1 billion years ago, another 600 million years ago, and about 250 million years ago, there were supercontinents that evolved. Let's see if I can actually get this link to work. I'm not so sure if I can do that. Here we go. So what this is showing is the supercontinent Pangaea breaking apart over millions of years into the world that we know today. Notice India is moving quite rapidly up to Asia, and then it's going to collide up with it to form the Himalayas, and that's the Himalayas are still growing because of this collision. All right, this cycle of rifting and patching repeats itself about every 450 million years. Very cool. So the supercontinents, what were they and how many were there? The earliest known supercontinent was Valbara. It was formed from protocontinents about 3 billion years ago. Valbara broke up also about 3 billion years ago, and then the supercontinent Kenorland was formed right after that, which broke up around 2.5 billion years ago into Laurentia, Baltica, Australia, and Kalahari. And then the supercontinent Columbia, or Nuna, formed about 2 billion years ago and broke up about 1.5 billion years ago. After that, the supercontinent Rodinia formed about 1 billion years ago and broke up roughly 750 million years ago. Plate tectonics brought the fragments of Rodinia back together in a different configuration during the late Paleozoic about 300 million years ago, forming Pangaea, which is the best known supercontinent. Pangaea subsequently broke up into the northern and southern supercontinents Eurasia and Gondwana about 200 million years ago. And it sort of looks like this. So that's Pangaea on the top left-hand corner, and that's at the Permian extinction. And then the Triassic, which is the time of the dinosaurs, 200 million years ago, and Jurassic, still time of the dinosaurs, and then Cretaceous, when the dinosaurs went extinct. Earth was sort of kind of like in its present form, and then today at the very bottom it is sort of like Cretaceous, except India has joined up with Asia. This is another version of the same timeline. At the bottom, you see the great big extinction at 250 million years ago that happened at the supercontinent Pangaea. And then about 100 million years ago, Laurasia and Gondwana land masses separated out. At the time of the dinosaur extinction, that's what the continents looked like. And then the collision of India with Eurasia happened about 45 million years ago, and we have what we have today, the configurations of the current continents. According to the theory of plate tectonics, Earth's crust is composed of plates floating on the Earth's mantle. And here is a link which will show you how this actually works. So this is kind of cool, because what you see here is the crust is being continuously formed here. It spreads out, and then it goes under another plate. It's subsumed by it. And here, this plate is just getting taller and bigger because mountains keep growing on it. Tectonic plates move slowly through the process of continental drift. Oceanic and continental plates collide, separate, or slide past each other, causing formation of mountains, islands, and earthquakes. Here are the major tectonic plates of the Earth. Not all of them, but some of them. And if you look at North American plate, it carries the North American continent, but not all of it, not California. And South American plate carries the South American continent. The Arabian plate is this one. It carries Arabia. The African plate carries all of Africa, and so on and so forth. Earth formed about 4.6 billion years ago. The early days were very hectic, with a lot of flying rocks and bombardment, so the surface was constantly vaporized and melted. So kind of hard to have liquid oceans at that time. But there is evidence of liquid water around 4 billion years ago. The early atmosphere was reducing due to all these eruptions of volcanoes, so it was mostly nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, hydrogen, all nasty stuff. When we think about the origin of life and we start to ponder on how could this have happened, is it even possible? So we start to look at very small things. Chemical and physical processes on early Earth may have been produced very simple cells through the sequence of somewhat plausible stages. First of all, there had to be abiotic synthesis of small organic molecules. And that's kind of possible because there is abiotic synthesis of amino acids in space. Then there had to be joining of these small molecules into macromolecules. That would need some addition of energy, but I suppose that could happen. Then the molecules had to be packaged into protocells. So we would have to make some sort of protocell, like a vesicle. And that is plausible because if you drop phospholipids in water, they self-assemble into a bilayer. And then the last one is origin of self-replicating molecules. That is the hardest one to explain away because there's some gaps in our knowledge on that. Amino acids have been found in space. Like I said, RNA monomers can be produced spontaneously. Small organic molecules polymerize when they are concentrated on hot sand or clay or rock. And clay especially because it's negatively charged. In water, phospholipids spontaneously self-assemble into a bilayer to form vesicles. And clay surfaces increase the rate of vesicle formation. There you go. So fluid-filled vesicles with a membrane-like structure could have formed protocells. Vesicles can maintain an internal chemical environment. And then we could actually go and replicate stuff. Replication and metabolism are key properties of life and may have appeared together in protocells. It could have happened this way. If you look at the top left-hand corner, we could have had this protocell, ancestral prokaryote, just cytoplasm and DNA. And then maybe it acquired endoplasmic reticulum. Maybe it acquired a nuclear membrane. Maybe it engulfed some bacteria, which were mitochondria. Maybe it engulfed some photosynthetic bacteria, which were actually plastids. The first genetic material was probably RNA and not DNA, and that is known as the RNA world hypothesis because RNA is single strand, DNA is double. But DNA is more stable and RNA is not so stable, so that would be better if it was the first genetic material because something that's not so stable we can sort of explain away. But even before RNA, there could have been something called TNA. TNA is actually triose nucleic acid, which is just like DNA and RNA. It can store genetic information in strings of nucleotide sequences. So it could have been. We got to DNA because DNA is more stable. RNA could have been the template, but TNA could have been the precursor for RNA. And then we come to extremophiles. So we've already somehow explained away how the first protocell could have formed, and now we're looking at these horrible environments that the early Earth was in, which were very bad. And what kind of life would have lived there? Those would be extremophiles. Extremophiles are archaeans which thrive in very hostile environments. They can live under immense pressure, as in the bottom of an ocean, where there's also scalding temperatures, maybe up to 172 degrees Fahrenheit near black smokers. And they don't mind great temperature changes. They move their heads from 72 degrees Fahrenheit to 172 degrees Fahrenheit. So 100-degree change while foraging for food, and they don't get into heat shock or cold shock. Here's a cartoon of extremophiles. So there are all kinds. Acidophiles, alkaliphiles, halophiles, Psychrophiles, Thermophiles. Acidophiles are organisms that live happily in acidic environments, which is below pH 3. And here's a picture of a pink New Zealand lake. And that pink stuff is actually acidophiles because the lake is acidic. And then we see Mount Vesuvius, which erupted some time ago. Archaeans called sulfolobulus living in the mountain. Psychrophiles are organisms that can thrive in very cold temperatures, 15 degrees or lower. And these are new ones, so we haven't quite identified them yet. So they're just given temporary names. Like, for instance, the one on the extreme left looks like Mickey Mouse. So for now, that Psychrophile is just Mickey Mouse. The one next to him looks sort of like a Klingon. The one at the very end looks sort of like leftover turkey. So we haven't identified them yet, but they're just given nicknames for now. Halophiles are organisms that live in saltier environments of at least 10% sodium chloride. So sodium chloride, 10%, is very, 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 very salty. If you look at this purple picture at the Great Salt Lake in Utah, that is in that much salt, these halophiles can live very happily. In the middle picture, salt flats along Highway 50 east of Fallon, Nevada, the pink stuff, that is halophiles living in the very, very salty water. And on the right-hand side, the orangey stuff, that's Owens Lake in Sierra Nevada in California. And there's a close-up of those halophiles, those little round blobs. And so they're happy there and they thrive there. Thermophiles are organisms that live in very high temperature, between 60 to 80 degrees centigrade. Those would be sort of like at the Yellowstone National Park in the geysers. But hyperthermophiles are extremophiles that live at 110 degrees centigrade. So those would be at in the black smokers, volcanic vents in the ocean floor. That's where they live. There are more kinds. There's alkylophiles, which are happy at alkaline environments of pH 9 or more, so that's more like bleach. Xerophiles, organisms that grow in environments with very low water. All life needs water at some point in its cycle, but these are not cactuses. Xerophiles are single-celled organisms, and they can live in environments with almost no water. Piezophile, organism that lives in very high pressure. Endoliths, organisms that live inside rocks. Hypoliths, an organism that lives inside a rock in a cold desert. Oligotrophs, organism capable of growth in nutritionally limited environments. Metallotolerant. So these would be extremophiles capable of tolerating high levels of heavy metals like copper, cadmium, arsenic, and zinc. There is a river in Spain called the Rio Tinto, which is the most polluted river in the world, and it has been like that for centuries. It's very acidic, and it's full of heavy metals like arsenic and so on. And in that river, it's teeming with our extremophiles. Radio-resistant, resistant to high levels of ionizing radiation. So they're extremophiles that don't mind being zapped. And polyextremophiles. Usually, extremophiles thrive at multiple bad conditions. They're bad for us, but for them, what we live in is bad. So it's really a matter of perspective. They usually like to have several kinds of bad conditions at one time. For instance, Archean endoliths have been found more than 4 miles down in the ancient lava flows of Columbia River area of the Washington state. These extremophiles live inside the rock itself and respire without oxygen. The pressure at this depth is about 70 tons per square inch. A car is about 2 tons, so that's like 35 cars stacked on top of each other, all squashing down this unicellular Archean. And he's very happy with that. We couldn't do that, but they can. There's also the notion of size. So how small can a gene be? How small a genome be? How small can a bacteria be? How small can an Archean be? There's some controversy over how small it can be, so the organism can actually boot up. But nanobacteria, which are very small, have been found at extreme depths at the seabed sandstone off Western Australia, and they survive at extreme pressures. Let's see if I can bring this up. This is kind of fun. It summarizes the extremophiles we were looking at.
Welcome to Bacillus infernus. New species of bacteria have been discovered in caves and gold mines as deep as 2 miles underground. These species are known as thermophiles, or heat lovers, because they thrive in high-temperature environments. The study of habitable environments deep below the Earth's surface may hold important clues to finding life on other planets. Many scientists believe that if there is life on Mars, it will be found underground. Hydrothermal vent communities. In 1977, scientists first discovered entire communities of organisms flourishing miles below the surface of the ocean, around openings in the ocean floor out of which hot, mineral-rich water erupts. The openings host a variety of strange and amazing organisms that have adapted to immense pressure, total darkness, and proximity to superheated water. There is strong evidence that a liquid ocean exists below the icy surface of Jupiter's moon, Europa. If this ocean contains hydrothermal vents as well, scientists speculate they could be well springs of alien life. Methane ice worms. In 1997, vast colonies of strange rosy pink worms 1 to 2 inches in length were found burrowing in mounds of methane-rich ice erupting from the seafloor in the Gulf of Mexico. The discovery of these previously unknown life forms living in frigid ocean depths far beyond the reach of sunlight extends our definition of what constitutes a habitable environment. The discovery provides further evidence that life is tough in the extreme. Methane ice worms.
Sorry about that.
Acidophiles. These hardy organisms have been found living in a number of caves under conditions about as acidic as battery acid. Some scientists speculate that the clouds of Venus could be a plausible habitat for microbial life. The clouds are highly acidic, but that wouldn't be a problem for acidophiles. Archaea strain 121. This microbe belongs to a class called Hyperthermophiles, which live in extremely hot or acidic water. Some species can survive temperatures over 235 degrees Fahrenheit, hot enough to boil an egg in minutes. The discovery of organisms that thrive within a broad range of temperatures opens up new prospects for finding microbial life elsewhere in the universe.
Let's see if we can get this last one.
D-radiodurans. D-radiodurans has been humorously dubbed Conan the Bacterium. It's the most radiation-resistant organism known. These guys can withstand 1.5 million units of radiation, 1,000 times more than any other life form on Earth. D-radiodurans beats most of the constraints for survival of life on Mars, including exposure to radiation, cold, vacuum, and dormancy.
Okay, so I thought I'd show you those. It's just what we've been talking about. There are extremophilic metazoa. So these would be multicellular organisms. And the first one I'm going to show you is the Pompeii's worm, which is this ugly looking thing at the bottom. And this is the one that hangs its head in cooler water and dumps his tail in scalding water. So we're going to take a look at this picture. It's going to zoom in. It's at the bottom of the ocean, near hot smokers. And here he is. That's his head. and he sticks it out. He's trying to cool it and look for food too, and then he goes right back in. And his tail is stuck in scalding hot water, 172 degrees, whereas his head is in cooler water. And then there's this guy. This is a tardigrade. This is in your backyard in the moss that you see. And I'm going to show you. So let's see if I can do this one. So they're also called water bears. Here's a better picture, I think. They look like little bears. You can't see them unless you have a microscope. And this is a scary one because what you might think is just moss might actually be a lot of these little guys moving around. Yikes, right? All right, and then there's the last one is the Antarctic krill, which is a crustacean, but it's kind of interesting to look at.
The freezing oceans around Antarctica are home to one of the world's most amazing crustaceans, the krill. Krill are usually found in large swarms out in open waters and are seldom seen by divers in Antarctica. But recently, during a dive in the Berthlow Islands, south of the famous Lemaire Channel, I was lucky enough to encounter a few adult krill swimming along a rocky kelp-covered wall. This gave me an opportunity to capture video of some of their behavior that has seldom, if ever, been filmed in the wild before. In the Antarctic food web, krill occupy the position that ecologists call a keystone species. Krill feed on diatoms, tiny phytoplankton, which bloom in fantastic profusion during the Antarctic summer. In turn, billions upon billions of krill provide a rich food resource for penguins, seals, and whales that may travel thousands of miles to take advantage of this bounty. Traditionally, biologists have thought of krill as plankton, a word which refers to plants and animals that are swept along by the ocean's currents, rather than swimming strongly against them. But some recent studies have shown that krill should be regarded as nekton, strong swimmers that go where they like fish and penguins. Krill swim in at least two important ways. In this shot, slowed to 1/3 speed, you can clearly see the amazing coordinated action of the pleopods, or swimming legs. Each of these 5 pairs of legs spreads into a paddle shape as it sweeps backwards, providing excellent propulsion.
Oops. Let's see if I can
This
get
is the
picture.
krill's normal method of swimming. Because they are heavy for their size, krill will sink to the bottom where they are easy prey for predators, unless they continue swimming virtually non-stop. In fact, krill have the ability to swim for weeks without resting. As it swims, the krill holds its tail, or telson, spread like a horizontal stabilizer. This tail can also be used for the second swimming method, a rapid escape.
All right, that's all I had for you, students. Goodbye.